Welcome back. So what we're going to do now is we're going to explore the second from our list of hypotheses as to what might be rate limiting for bacterial growth. Before I start doing my actual calculations, let's just revisit what we had to say about the matter during the last vignette. So as you can see here, we started out with this hypothesis of the rate of sugar uptake as being rate limiting. And after carrying out some calculations, we showed that it took on the order of several thousand sugar transporters. And there's certainly enough room for the cell to add more transporters in order to accommodate higher rates of uptake. So now what we're going to do is we're going to think about the rate of ATP synthesis. And in this case, the idea is that it takes a certain number of ATP hydrolysis events in order to carry out the processes of the cell associated with cell division. And what we want to do is we want to try to work out how many ATPs that is and what that implies about the number of ATP synthases. Okay, so just a quick reminder that if you recall, ATP is often noted as the energy currency of the cell, and here I just simply wanted to show you that the chemical structure is this nitrogenous base, and then there's a sugar, and then there's three phosphate groups. And the hydrolysis of this thing leads to a free energy release that's used for a myriad of different reactions within living cells. And what we're going to do is we're going to focus on only one of those, and that is the process of protein synthesis. Now, my reason for that is that, yes, there are certainly more processes than just polypeptide bond formation, but this is actually the majority player. So it's something, if I remember correctly, like 85% of the budget. And so what we want to do is we want to try to understand each polypeptide bond is worth 4 ATPs, and we're going to explore the implications of that. So at any rate, what I think I have to say now is best served by going over to my iPad. And so here, as you can see, the idea is that there's a molecular machine, the so-called ATP synthase. It lives in the cell membrane. It rotates. It's actually a rotary engine. It rotates at around 6,000 RPMs, which, in case you don't have an intuition for that, that's basically the number of RPMs that are associated with the jet engine at the end of the runway when the pilot revs it up for takeoff. So 6,000 RPMs, which is 100 hertz, 100 rotations per second. And as I show you here, basically every time that the ATP synthase makes one complete rotation, it produces 3 ATPs. So what I would say that we learn from that is that there's roughly 300 ATPs synthesized per second. So let's note that here. So I'm going to say 300 ATPs per synthase per second. So that's the intrinsic rate at which these guys operate. So what I want to do now is I want to try to come to terms with how many ATP synthases are there. And let me actually add a page here. So good. We have a page, and so now I can try to figure out how many ATPs need to be generated for one cell division. So that's the question that we want to address. And the logic, just as a reminder, I'm intentionally ignoring the fact that there are other processes, and the logic is we're going to focus only on protein synthesis. And the claim I'm going to make is that there are 4 ATPs equivalents. Let me be a little bit more precise here. So 4 ATP equivalents per peptide bond. reason I say equivalence is because really it's GTP hydrolysis and so on, but the reckoning is basically the same. So how many polypeptide bonds per cell division? That's the first thing we want to figure out. And the way that we're going to attack that is we are going to remember, so the number of proteins per cell we said was something like few times 10 to the 6th. And the number of amino acids per protein is of order few times 10 to the 2. So this implies, that's my symbol for it, implies, this implies that the number of polypeptide bonds is given by F times 10 to the 6th proteins times F times 10 to the 2 amino acids per protein times 4 ATPs per amino acid, and this ends up giving F times F is 10, so using few times few is 10, then this leaves me with 4 times 10 to the 9th ATPs per cell division. So that's our estimate for the number of ATPs that need to be synthesized. And now what we want to do is we want to try to figure out what that implies about the number of ATP synthases. So how many ATP synthases are required to do that? In, let's say, F times 10 to the 3 seconds. All right, so I have a number of synthases is equal to 4 times 10 to the 9th ATPs divided by F times, oh wait, one sec, so F times 10 to the 3 seconds, so that tells me that, and then so that's the number of seconds in the cell cycle, and then the final factor is going to be 300 ATPs per synthase per second. So that gives me the right units, which is synthases, and I see that is, so let's be careful, 4 times 10 to the 9th divided by F times 3 is 10, so this is 10 to the 6th, so this is of order 4,000 synthases. So that's our conclusion. We conclude that on the basis of this simple estimate, we conclude that the number of synthases is of order 4,000. So let's see now how this jibes with what was in our recent paper, which you can find on the archive by Griffin Chure and Nathan Beliveau. So they did the estimate for 5,000 seconds, as you can see down here at the bottom right of this little pink box. And here what they're plotting is the brown dot at 0.5 growth rate per hour is their estimate, which they said was on the order of 3,000, and we found on the order of 4,000. And what you see is the trends as a function of growth rate. What do I mean by that? Well, what I mean is that the number of ATP synthases in the cell, as measured by both ribosomal profiling and by by mass spec is an increasing function of the growth rate. So the faster the cells are growing, the more ATP synthases per cell that you will find, not surprisingly. And and so, what I would say is that once again, this forms a very coherent picture. It's coherent in the following sense. What we found is that the number of ATP synthases required is on the order of several thousand. Okay, I don't want to put too fine a point on it. And remember that I made the remark, and I'm going to come back to this in more depth in another vignette. The membrane is decorated with proteins to the tune of roughly 100,000 membrane proteins per micron squared. And there's around 5 or 6 microns squared in the bacterial membrane. And so clearly this is just a drop in the bucket in the sense that the ATP synthases are taking up hardly any of the overall real estate of the membrane. There's plenty of room to add more ATP synthases. And so once again, we end up rejecting this hypothesis, quote unquote hypothesis, as to a possible rate limiter, because what we found is that the ATP synthases can easily accommodate the energetic demands of the cell. It won't make any difference if you worry about, for example, how much does it cost to do DNA synthesis or lipid synthesis or ribosomal synthesis or whatever. It's not going to change the story much. Stated differently, the thing that I would say is that you can basically, as a rule of thumb, say that cellular life demands something on the order of between 10 to the 5th and 10 to the 6th ATPs per second per micron cubed of cytoplasmic material. That's the rule of thumb that I'm carrying around. And you can see that means you need several thousands of ATP synthases to keep up with those demands. So several thousands of ATP synthases per micron cubed of cytoplasmic material. Now, in another vignette, we're going to talk about the surface to volume ratio and how this becomes tricky. But for the moment, that paints the picture. So to summarize, that's the end of this vignette. To summarize, we've now addressed 2 of the competing hypotheses for what limits bacterial growth. We've considered sugar transport, and we've considered energy generation in the form of ATP synthase. And we found that both of those hypotheses are wanting because we see that the cell is easily able to keep up with both of those demands by simply adding more proteins to the membrane, either to transport sugars or alternatively to generate ATP synthases. Where we're going in the future, next we'll briefly consider a vignette in which we think about cell wall synthesis, lipids in cell wall, and then I'll have a separate vignette on membrane area and the surface to volume ratio, because I think that's a fun little detour. It'll be brief. And then what we'll do is we'll turn to the processes of the central dogma. How much does energy and how much ribosomal content is required to keep up with protein synthesis? How long does DNA replication take? How long does transcription take? And so on. So with that, we wrap up this particular